Today we're going to talk about Chapter 14, and Chapter 14 is the start of our immunology lectures. Okay? So 14 and 15 deal with two parts of the immune system. Today we're going to talk about the innate immune response, and then in 15 we'll talk about the adaptive immune response. Okay? The immune system is relatively complex. There's a lot that's going on. So don't get overwhelmed. Don't freak out if you don't understand every single thing that I'm saying. Okay? What I'll do at the end of both of these is I'll kind of summarize it into a nice little short and sweet story. Okay? That makes more sense, hopefully. So let's get into it. So what we're going to do over the next couple days is talk about the host defenses. Okay? Host defenses is really just a generic term for anything your body does to prevent infection. Okay? This could be physically or chemically blocking the organism from gaining entry into the body altogether, or it could be utilizing the immune system. Okay? And the immune system are a series of cells, which you can see here down in the yellow panel, that circulate and migrate around the body looking for anything that is deemed bad, okay, or what we call foreign. And if it is bad, then these cells are there to eliminate it, hopefully destroy it, and then help you prevent that infection, or help you get better if you've already been infected. As I said, this is classically broken down into two different systems, the innate immune response and the adaptive. We'll talk about innate today and then adaptive more in 15. And then as I said, it's really 3 lines of defense, or at least this is the way your book categorizes it. And you have your first line, your second line, and your third line. Okay? The first and the second line fit in within the innate immune response. And the first line of defense is literally just physically or chemically blocking the organism from gaining entry. Okay? So this really isn't classically considered part of the immune response. Instead, it's just a way to stop it from infecting, or stop the pathogen from infecting you altogether. Traditionally, when we think of the immune system, we think of these last two over here. Okay? The second line and the third line, or the acquired immune response. We're going to focus on this guy today. And there's 4 branches of the second line of defense that we'll hit heavily today, and that's inflammation, interferons, phagocytosis, and complement. And so, what is the innate immune response? So the innate immune response is usually the first immune response that develops during an infection. Okay? And that's because these cells are literally scattered throughout the body into most of the tissues in the body. And they're literally just sitting there waiting for an attack. Okay? So we say they're always on guard. They're literally all over the place, and they are just sitting there waiting for something to occur. Now it's important to note that this is also known as the nonspecific immune response, and that's because these cells tend to respond the same way to all pathogens, okay, for the most part. So if they see a bacteria, they don't care if it's E. coli or if it's S. aureus or S. pyrogenes, whatever, it will respond to that bacteria in a very similar manner. And their primary goal is to phagocytose, so get rid of that organism if they can, as well as start something called inflammation. And inflammation is what we call the hallmark of an immune response, which means it pretty much always happens whenever your immune system is triggered. And we'll talk more about why that is, but it's actually a pretty beneficial thing because it attracts other immune cells from other parts of the body to go to that area. So as we mentioned before, the first line of defense is really not part of the immune system, at least classically speaking, but they do help prevent infection. Okay? And this is by using an anatomical or physical barrier, or physical blockade, a chemical barrier, which is releasing a chemical that destroys the microorganism before it gains entry into the body, or a genetic barrier, which is just having good genetics and makes you a little bit stronger. Maybe your skin's thicker, or your immune system responds better, or something along those lines. So when we think of physical barriers, these are thick, kind of rough tissues that are designed to prevent organism from gaining entry into the body. When we often think of this, first thing that comes to mind is going to be the thick skin. We've already kind of talked a little bit about this. Your skin has a couple layers of dead keratinized skin cells. These are very hardy and rough and difficult for most pathogens to break through. Okay? So it's a very good way of preventing the organism from getting into the body at all. But there are other physical barriers, so mucus is an example. This is a slimy, kind of sticky liquid found at mucous membranes. And it's designed to catch pathogen, and then your cilia will actually propel it up and out of the body. Okay? So the name of the game for physically blocking is just either stopping it from gaining entry or moving it out of the body before it can cause infection. Chemical defenses. So chemical defenses are releasing some sort of chemical in the body that destroys the microorganism. Okay? The classical one is something called lysozyme. This is an enzyme that is released in mucous membranes as well as in tears, okay, or in your eyes. And it literally chews through the cell wall of bacteria. This will then kill the bacteria and prevent the infection. Okay. Some other examples, so sebaceous secretions or oil secretions on the skin make it somewhat inhospitable for a lot of organisms to survive, as well as the lactic acid, electrolyte concentrations, and even the acidic pH of the skin. Okay? So not only is the skin a great barrier, there are also a lot of chemical defenses on the skin to aid in this protection. Okay? And this just goes to show you how important the skin is at protecting you from becoming infected. Within the digestive tract, you have hydrochloric acid in the stomach. This is a pH of around 1 to 2, which is really caustic. And so a lot of organisms will die in this acid before they cause disease. Digestive juices and bile, again, these are used for breaking down foodstuffs, but you can also break down potential pathogen. And then antimicrobial chemicals and semen, which are often peptides, as well as the acidic pH in the vagina. So having these physical and chemical barriers are a great place to start, but pathogens evolve very quickly, and most of them have evolved ways to get around all of these things. Okay? So yes, these are there, and yes, they help, but they aren't perfect. So this is why we need to have an immune system that's there to protect us in case these fail. And then, as I mentioned before, genetic differences can also determine how susceptible you are to getting an infection. Maybe you have thicker skin because you have the genetics for it. Or your immune system that makes more neutrophils than your neighbor, and so you fight infection a little better. Okay? So genetics also does play an important role with this. Okay, so now let's actually get into the immune system proper. Okay? As I said, today we're going to talk primarily about the second line of defense, known as the innate immune response. And in this we'll have inflammation, interferon, phagocytosis, and then complement. And then in chapter 15 we'll deal with the third line, also known as the adaptive or the acquired immune response. And this immune response is the immune response that really kicks into gear when infections get really bad. Okay? So innates are the first guys to come in. If they can keep it under control, that's where it stops. But if it starts to get out of control, then the third line will kick in, and these are the big guns. These are the guys that can come in and really do a lot of the clearing of the pathogen. The third line of defense is really, really specific. So I told you earlier with the second line, they kind of respond to the same bacteria the same way every single time. Well, with the third line, it's very, very, very specific. They can tell differences between very, subspecies of certain bacteria. Okay, so not even all E. coli looks the same to the third line of defense. The way the third line of defense works is by production of antibodies, B and T cells, and we'll talk more about what those exactly do in chapter 15. So what is immunology? Well, immunology is the study of how your body defends itself. Okay? And it's primarily by looking at immune cells, also known as white blood cells. And these are cells that circulate around in the lymphatic system, the bloodstream, as well as in tissues, and literally look for any abnormalities. Okay? Whether that be your own cells that are growing uncontrollably, like in a cancer, or it be some sort of foreign entity like a bacteria, a parasite, virus, or what have you. So their main goal is to survey the body. They literally go into every nook and cranny, except for a few cases, of the body, looking for anything that is strange. Once they are there, they then have to recognize what is foreign and what is not, or what is strange and what is not. And then once they do that, they need to destroy it. Okay? So their job is relatively difficult. It's relatively complex. And that's because these are just a whole bunch of loose cells. Okay? It's not just one organ that's doing all of this for you. It's literally billions of cells that are just migrating all over the body constantly, looking for anything that is weird. Now, for them to survey the body properly, they need to understand what is self and what is non-self. Okay? They need to be able to look at a cell and be like, you know what, you're cool, I'll let you go. You know what, you're weird, I'm going to kill you. Okay? And it really boils down to what is expressed in that cell, or what we call markers. These markers are specific proteins or specific molecules that are found within cells that aren't normal. Okay? So an example of a marker would be something like LPS, lipopolysaccharide. This is a molecule found only on gram-negative bacteria. So if an immune cell goes up to it and it sees LPS, it goes, you're a bacteria, I'm killing you. Okay? It's looking for these unique kind of markers. Now, the adaptive immune response, which we'll talk about in chapter 15, can take it even a step further. They can actually discern what cells are from you and what cells are from you, right? So it's not even if they're human or not, it's just if they are actually yours. And this is why if you get an organ transplant, if you don't, curb your immune system will destroy it relatively quickly. So where do we find immune cells? Well, as I said, there's no real organ per se that they're in. Instead, they're littered all over the body. Okay? So you find them in places like the RES, the reticular endothelial system, which is basically kind of the open space between cells and tissues. Extracellular fluid, which is the fluid found within this open space. The bloodstream, which is the cardiovascular system, and the lymphatic system. By far and large, most of your immune cells are found within this bloodstream lymphatic system. Okay, so that's what we're going to focus on today. And classically speaking, this is where you think of most immune cells. Okay? Now, because these cells are littered throughout the body, they need to have very good ways of communicating with each other. If they don't communicate with each other, then it's just a bunch of little cells willy-nilly all over the place. Okay? They need to communicate so that if there's a problem, they can send out a signal and all the immune cells go to that one area and help fight the infection. They do this primarily by releasing proteins or chemicals. Okay? And we call these cytokines. And we'll talk more about what those are in chapter 15. Now, as I said, classically, when we think about immune cells, we often think of them in the bloodstream or the lymphatic system. Okay? We're going to primarily talk about the lymphatic system because this is the area where, during an infection, most of the immune cells are going to be communicating in. Okay? This is the place they like to be for the immune system to really kind of take off. So what is the lymphatic system? Well, it's a series of vessels and nodes, okay, that basically run parallel to the cardiovascular system. They are very thin walled, so they look a lot like veins, okay? And their primary purpose, at least from a physiological point of view, is to collect extracellular fluid, okay? So whenever your heart pumps in the cardiovascular system, occasionally some of the liquid from the blood or plasma will leak out. And it's the lymphatic system's job to kind of sop that up and then bring it back to the core of the body so it can be recycled and reused, okay? However, the lymphatic system also plays another relatively important role, and that is the surveillance, recognition, and protection of foreign materials. A lot of your immune cells use the lymphatic system as a place to not only get around the body, because it's basically a network or a highway they can use, but also they use it as a place to communicate. Okay? So when you look at the lymphatic system, which is right here, you can see it's a series of vessels, okay, or kind of tubes, that look like veins. But there's also these little pockets, or what we call nodes. Okay? These are little openings in the vessels, and this is a place for immune cells to kind of hang out and talk to each other. And the real beauty of the lymphatic system is that it moves under very low pressure. So if you think of the cardiovascular system, your heart's constantly pumping and it's just pushing shit around really, really fast, right? In the lymphatic system, there's no muscle present that pumps it directly. Instead, the only thing that moves lymphatic fluid is movement of skeletal muscles. So every time I move my skeletal muscles, it puts a little bit of pressure on these vessels, and this pumps the fluid towards the core of my body. Yeah?
Why do lymph nodes get swollen?
So lymph nodes get swollen during an infection because let's say I got infected on my hand, right here. All the immune cells that are hanging out there will get into the lymphatic system, They'll go down this lymph vessel until they hit the next biggest lymph node. They go to that lymph node, and that's where they all kind of hang out, talk, and then they start to expand and grow. And when that happens, the node gets bigger and bigger and bigger. So it's a sign of an infection. And where it is, whether it's in your neck or in your armpit or in your groin, it gives you an idea of where the infection is coming from. Because they literally go to the first one that they see. Okay? Now there are valves located within the lymphatic system, which means fluid does only want to run in one direction, and that is from the extremities to the core of the body. And that makes sense, again, because it's designed to collect excess fluid, and you would want that to go into the core of the body where it then can be recycled. This is one reason why if you are bedridden, like in a hospital for example, you tend to get a lot of edema or buildup of fluid. It's because you're not moving skeletal muscles, you're not pumping the fluid through your lymphatic system, and the fluid just pools in those tissues. So what is lymphatic fluid or lymph? It's basically plasma. Okay? Plasma is the clear part of blood. Okay? It's basically water with some other proteins like albumin. And it's not surprising that it is very similar to plasma because, as I said, the lymphatic system is designed to collect the excess plasma that leaves the cardiovascular system. So really it's just, water with a little bit of protein in it. The lymphatic vessels, as I said, they run relatively parallel to major arteries and veins, blood vessels. They're very thin-walled, so they look a lot like veins, and you have most of them in your extremities. Again, it's because it's designed to drain excess fluid from the extremities. But it's also important to have a lot of these in the extremities for your immune system. Because where are you most likely going to get cut or get hurt? In your extremities, right? You don't often get cut here unless you get stabbed or something like that. As I said, the flow of the fluid runs in one direction, from extremities towards the heart. And again, it's only moved through the vessels through contraction of skeletal muscles. Now, when we look at the immune system, there are some organs or tissues in the body that are designed to help the immune system along. Okay? They're not just only immune system organs, but they are there to help. We categorize these into two groups, primary and secondary lymphoid organs. Primary lymphoid organs, this is where we actually make or mature lymphocytes or immune cells. So an example of these would be the thymus, which is used for making T cells, and the bone marrow, which is used for making all blood cells. Okay. The other category of lymphoid organs are the secondary lymphoid organs, and these are the organs that are used to allow for the immune system to do their jobs. Okay? So the cells are already made, but this allows them to get around the body and communicate and all that kind of stuff. You have the spleen, which is used in the bloodstream to collect immune cells so they can communicate with each other. And then you have the lymph nodes, which we've already talked about, are little openings in the vessels for immune cells to communicate. So again, lymph nodes, these are small bean-shaped organs. They do get larger, as you said, during an infection because there's more cells in the area. And they're literally there just to allow for cells to stop and pause and talk. Okay? Being able to communicate, or at least for the immune cells, is very, very important for their function. So they need to have a place where they can take a breath and just kind of talk with each other. Where do we find these? We find most of them in the axillary, inguinal, and cervical nodes. Okay? So around the shoulders, around the extremities and the leg, as well as around the gut. Okay? And again, this is where you're most likely going to come into contact with potential pathogen, because this is where you're most likely going to get a cut, a scrape, or something along those lines. The spleen, so this is basically a lymph node in the bloodstream, okay? It's a relatively large organ, okay, and its job is to collect fluid, the blood, and in it there will be immune cells which then can talk with each other and things like that, okay? Without this, you become more susceptible to getting bloodborne infections, okay? And it takes you a little bit longer to clear an infection than it would with one of these, okay? But you can have your spleen removed if it's infected or something weird, and you live a relatively normal life. The thymus, as we mentioned before, the thymus is an organ that's used to produce T cells. Okay? We'll talk more about the thymus in chapter 15 when we talk about T cells. But it's a relatively large organ at birth, And then as you get older, by the time you reach about puberty, it atrophies or shrivels up to about the size of a walnut. Okay? Its main purpose is to teach T cells how to function, how to work in the immune system. And the reason why it shrivels up by the time you reach puberty is because by that point you've pretty much made all of the T cells you're going to have for the rest of your life. Okay? And now there are a lot of other lymphoid tissues. We're not going to talk that much about these. Really, these aren't just for the lymphatic system. Instead, they are just kind of areas of the body that have more white blood cells than other parts of the body. Okay? So some examples would be tonsils. These are basically big lymph nodes in the back of your throat. Breasts of pregnant women, because there's a lot of lymph nodes in that area, and you can get a lot of immune antibodies and things that are transferred to the child. And then galt, malt, salt, and balt. All of these are just lymphatic tissue found in different areas: the gut, the mucosa, the skin, or the bronchial areas. All right, so that is the lymphatic system and where the immune cells like to hang out and kind of move around. Now let's actually talk about some of the cells and what they do. Okay? So all blood cells, whether they be red, white, or platelets, are produced in bone marrow through a process called hematopoiesis. Okay? This is where you have stem cells found in the bone marrow that can differentiate or turn into really any kind of blood cell the body desires. Okay? They need red blood cells, they dump some chemicals on the stem cell, and bing, bang, boom, you got some red cells out of it. So red blood cells, also known as erythrocytes, white blood cells, known as leukocytes, and platelets, known as thrombocytes, all are produced through this hematopoiesis process. And again, it's based on something called differentiation, which is basically where one cell can differentiate or turn itself into many, many, many other types of cells. And what determines this is the chemical environment. So if the cell needs red blood cells, it dumps some chemicals on hematopoietic stem cells, bing, bang, boom, you get red blood cells out of it. Okay. Now, we're not going to talk too much about the red blood cells or the platelets because they don't really help the immune system much. Instead, we're going to focus on the guys in the yellow. These are the white blood cells or the immune cells. So when we look at the white blood cells, we can break them up into two categories: granulocytes and agranulocytes. The reason they have this name is because granulocytes have granules in them, okay, little dots. So when you look at them under the microscope, you see a bunch of little dots. Agranulocytes do not have granules. When you look at them under the microscope, there are no dots. Okay? Within the granulocytes, we have neutrophils, basophils, and eosinophils. And within the agranulocytes, we have T cells, B cells, and monocytes. For the innate immune system, what we're talking about today, that's going to be the neutrophils, basophils, eosinophils, and the monocytes. Okay? The T and B cells are going to be more in chapter 15. So let's start off with the granulocytes. First and foremost, we have the neutrophil. Okay? The neutrophil is by far and large the most abundant leukocyte. Okay? They are also very short-lived. They only live for a couple weeks at a time. Their main job is to phagocytose. Okay? These are often the very first cells to come at the site of an infection, and their job is just to literally gobble up anything and everything that is bad or foreign. I often call these guys the garbage trucks. Okay? They gobble up anything that is foreign. Now that's literally all they do. They gobble it up, and then a couple hours later, they die. Okay? They do not live long. They do not help any other part of the immune system. They literally are just there to clean up the garbage. Okay? In fact, whenever you get pus, so like in an infected cut, all that white kind of fluid, that is actually dead neutrophils. That's how many neutrophils go to the area to fight the infection and then just quickly die. Eosinophils. So eosinophils are also innate immune cells, and these are the guys that are designed to get rid of large parasites. They are large pathogens. So you can imagine that if I have a large worm in me, let's say it's a couple centimeters in length, there's no way a little baby neutrophil is going to be able to phagocytose it and get rid of it, because it's just too big. So instead, the eosinophils will come in, and what these guys do is they create a chain of cells that completely circles around that large parasite. They then dump nasty things on top of them. Okay, they degranulate. And this is supposed to annoy the parasite so that it latches off and falls off the body. Okay? In theory, it sounds like a good idea, but most of the time it does not work very well. And that's because these parasites have developed ways to get around the nasty chemicals. And what it ends up usually doing is causing eosinophilic granulomas, which are big kind of balls of scar tissue that develops around these cells. Okay? And if that happens in the brain or in the liver or other places, it can cause tissue damage. Now, if you live in a first world country like we do, where there aren't many parasites, these eosinophils get bored. Okay? And when they get bored, they start doing things they shouldn't, and that's primarily causing inflammation, random inflammation, as well as allergies. So these cells are important in causing allergic responses when they get bored and have nothing better to do. Okay. Basophils, this is the cell, or the type of cell that's in lowest quantity within the circulation. And its job is to create mast cells. Mast cells are cells that are really important in causing allergies, just like we talked about with eosinophils, but also causing inflammation. Okay? So they're bad in the fact that they cause allergies, but they are the guys that start the inflammatory process during an infection, and this is what recruits immune cells and helps you clear the infection at the end of the day. Okay? So they're both good and bad, if you want to think of them that way. Now onto the agranulocytes. So as I said, we have T cells and B cells. These are what we call lymphocytes. And these we'll talk more about in chapter 15. Okay? They're kind of the brains of the operation. They're the ones that either orchestrate the immune response if it's a T cell, or if it's a B cell, make antibodies. Okay? The other agranulocyte are monocytes. And monocytes are part of the innate immune system, so we're going to talk about those guys today. These guys are really good at becoming tissue macrophages, and all these cells do is really phagocytose. So they're really good at gobbling up foreign material. So a little bit more about monocytes. So monocytes are produced in the bone marrow. They go into the blood. And when they're in the blood, we call them monocytes. But as soon as they leave the cardiovascular system and go into a tissue, they then are called macrophages. Okay? So the name just tells you where in the body they are found. Now, as I said, their primary, and also it's important to note that they are very long-lived. They can live literally for your entire life. Okay? They get produced in tissues and they just sit there for decades, okay, until something kills them. So their main purpose is to phagocytose. So they go in, they gobble up, they kill bacteria, viruses, and small parasites. But not only can they kill, like neutrophils can, they can also process whatever they kill and present it to other immune cells. And this becomes really important when we talk about chapter 15 and the adaptive immune response. Because the adaptive immune response can only get turned on during or using this process. If the macrophages aren't there and they aren't processing and presenting, you will never get a third line of defense. Okay? You need these cells to be able to bridge the gap. So not only are they garbage trucks, they're also kind of like policemen, right? They kind of dictate what's going on and what's going to happen based on what they show. Okay? And then once they do find something, they can secrete compounds like cytokines that then activate and turn on the rest of the immune system. So they serve a very important role. Okay? Now macrophages can also become what we call dendritic cells, and these are cells that are basically really good at the second point. Okay? That's all they really do is present. So here's an example of a macrophage up at the top. You can see all these little pseudopods, or little kind of arms that are coming out of it. And these are designed to grab whatever is around them. Okay? So this guy's sticking his little arm out, he's grabbing a bacteria and pulling it into him, and so he can break it down and do whatever he needs to do with it. Now, these guys, as I said, once they leave blood, they go into tissue, they become macrophages. And depending on what tissue they are in, sometimes they get different names. Okay? So in this example, we have a liver. This is liver tissue. You can see the hepatocytes, which are these orange-looking things. And then next to them, these big kind of pink blobs, those are Kupffer cells. And Kupffer cells are basically macrophages of the liver. Okay? Down here we have some skin, so you can see the epidermis and the dermis, and you see these like weird looking pink things with arms. These are called Langerhans cells, and these are macrophages of the skin. Okay? They all are very similar in the way that they work, except because they're found in different tissues, we give them different names. Now, hematopoiesis is also used to produce erythrocytes or red blood cells and platelets or thrombocytes. But again, these don't really help too much in the immune system, so we're not going to talk too much about it. Erythrocytes are there to transport oxygen and remove carbon dioxide from tissues, as well as transport nutrients. And platelets are used for clotting, okay, or creating kind of a sticky mass that blocks blood so that it doesn't, you don't bleed out to death. All right, so now let's talk a little bit more detail about those 4 processes we talked about within the innate immune response. Inflammation, phagocytosis, interferon, and complement. Okay? And as we said, these are often called the non-specific immune responses because they respond pretty much the same way to all pathogens. Okay? It doesn't change too much. So first off, we'll talk a little bit about inflammation. So when you hear the word inflammation, most people think it's a bad thing. Okay? In reality, inflammation is really important for the immune system to function properly. As I said before, we often call it the hallmark of the immune response because without it, the immune response wouldn't happen. Okay? So it always occurs just in low levels. Now, obviously, there can be too much of a good thing. And so if you have really the widespread inflammation or it lasts for a long period of time, then it can cause problems. Okay? But a little bit of inflammation during an infection is very important. So what is inflammation and how is it brought on? Well, inflammation starts through some sort of tissue damage or destruction. So whenever your tissues are destroyed, whether that is by yourself, by something you did, like getting pricked by a rosebud or rose bush, or something that a pathogen is doing, releasing exoenzymes and toxins that are destroying tissues. All of those will result in the same thing, and that is inflammation. Inflammation is basically redness, warmth, swelling, and pain, and sometimes loss of function within that given tissue. The most telltale sign of inflammation is the swelling, also known as edema. And this is where your blood vessels in the area become leaky, and so a lot of fluid will start to leak out of them, and this creates a lot of liquid within that tissue. Okay? So why have inflammation? Why is this beneficial for the immune response? Well, first and foremost, as soon as you get inflammation, it tells immune cells where to go. Okay? So if I cut my finger right here, that will cause tissue damage. That will cause inflammation. And then all the immune cells down in my feet will migrate up, and they'll go to where that inflammation is. And if you think about it, it's relatively genius in the way that it works. Because where are you most likely going to get an infection at that point in time? Where you cut yourself. Okay? So this inflammation is what attracts immune cells to the area. Now, not only does it attract immune cells, it also starts mechanisms for repair, like release of platelets, as well as repair mechanisms and repair cells. And if there are any microbes that come in during that tissue damage, you're also going to get the immune cells present, and they can help fight that infection. So here's just a nice little cartoon diagram of a basic inflammatory process. Okay? So up at the top in number 1, we've got a needle, and this needle is covered in E. coli. Okay? This person takes the needle and jabs it into their arm. By doing this, they are destroying tissues and cells in that area where the needle goes in. And this causes mast cells, which are littered throughout your body, all over the place, to degranulate. And all that means is they release chemicals. Okay? These chemicals, what we call chemokines, start the inflammatory process. Okay? And the first thing that occurs is your blood vessels will dilate, okay, become bigger, and a bunch of fluid starts to leak out of them. Okay? And this is what brings on that edema or buildup of fluid in the tissue. Now these chemicals also create a gradient, okay, within the bloodstream, and this allows neutrophils to sniff that out and find where it came from, in this case where the inflammation has occurred. So it attracts neutrophils to the area, at least initially. These neutrophils then come in and they gobble up as much bacteria as they possibly can. In the process, this is where you develop pus because the neutrophils start to die. Okay? Now, if for whatever reason, these neutrophils cannot get all the bacteria under control, then more immune cells will start to come into the area. The next ones will be the macrophages and the lymphocytes, which then can step up the immune response and make it even more potent to help fight the infection even better. Okay? So inflammation is important because it starts all of these processes. It's what brings the immune cells to that area to help fight the infection.
Why would someone infect themselves with E. coli?
Oh, I don't know. It's just the example I had. I mean, anytime you jab yourself with a needle, you're potentially infecting yourself. But yeah, because they're an idiot. All right, so what brings on inflammation, as I said, it's chemical mediators. These chemical mediators, often called chemokines or cytokines, these are just proteins that are released whenever inflammation needs to start. Okay? So tissue damage, these chemicals are released, bing, bang, boom, all the cells in the area start to cause inflammation. And then these chemokines that are released act as a nice little kind of signal for white blood cells to follow, which eventually leads them to the site of that inflammation. And as these build up, they can cause things like fever or also stimulate other immune cells like white blood or lymphocytes via T cells. And they can also prevent virus spread and even sometimes cause allergies in certain people. Okay? So they are relatively potent and can do a lot. So what exactly is edema? So as I said before, edema is essentially buildup of fluid in a tissue. Okay? And this is brought on by inflammation, which opens up the blood vessels and causes them to leak out fluid. Okay? This leaked out fluid puts pressure on the tissues and kind of opens it up a little bit, and that's what causes that swelling in your infected or inflamed tissue. Now, it does this so that two things can occur. First and foremost, something called diapetes, also known as extravasation. And this is where cells leave the blood vessel and go into that tissue. Okay? So by having that inflammation, it allows these cells to easily wiggle through and get into that area to help fight off anything that may be in there. And then it also allows for chemotaxis. We've talked about chemotaxis before with bacteria in chapter 4. And this is basically sniffing out a chemical gradient until you find the source. Okay? In this case, instead of it being shit like we talked about with bacteria, it's now chemokines and cytokines that are released during inflammation. So here's an example of diapetes, extravasation. You've got neutrophils in the bloodstream, right? As soon as you get inflammation, there are chemical signals that are released. Okay? These cause the blood vessels to open up. Fluid comes through, okay, into the area, and this causes edema, okay, and inflammation. This also allows the neutrophils to push themselves through these little gaps so that then can enter the tissue and start to fight off the infectious agent. Okay? This is what it looks like in real life. So here's an electron micrograph, here's your blood vessel, and you can actually see this little guy pushing himself through to get to the other side. So what are the benefits of having edema and chemotaxis? Well, if you have an organism that produces a toxin, this extra fluid will dilute the toxin, okay, making it less potent. Having this edema also brings in clotting factors, which can clot the blood, prevent you from bleeding to death, which is good. It makes it easier for phagocytosis to occur, because now there's more volume in the area, you can fit in more cells to help fight the infection. And as we said before, sometimes it will result in pus, and this pus is where you have a bunch of neutrophils that are going crazy and then eventually die. Now, as I said, having a little bit of inflammation and edema is a good thing. And if your immune system, or in particular your neutrophils, can clear out all the pathogen or potential pathogen in the area, then you're fine. Okay? However, if they can't, then this inflammation will continue, will build up a little bit, and this then recruits in other immune cells that can kind of step up the response. Okay? Next will come in the macrophages, And these guys will clear the pus, cellular debris, dead neutrophils, damaged tissue, as well as any bacteria or pathogen that is still there. They can then gobble that up, process it, and present it to the B and T cells, which are important in the production of antibodies, as well as kind of making sure the immune system goes into full-blown, full-blown attack mode. Now, obviously, if this occurs, inflammation occurs for a very long period of time, then you can actually develop scar tissue and problems with the tissue, and so that's usually not a good thing, okay? But again, having a little bit of inflammation for a little while is probably better than not. All right, so that is inflammation and the inflammatory response. Now let's talk a little bit about phagocytosis. So we've talked about phagocytosis pretty much all throughout the semester. And this is where you have specialized cells called phagocytes in your body that gobble up anything that is bad, okay? Whether that is a foreign agent like a pathogen, a virus, or a small parasite, or it could even be your own cells that are being abnormal and weird like in cancer, okay? Their primary purpose, these phagocytes, is to survey the body and look for abnormalities. Once they find it, they then ingest whatever that abnormality is, and then break it down and eliminate it, okay? And if you are a macrophage, you can then also process this material and use it to turn on the immune system later on, okay? The T cell. There are three main types of phagocytes: neutrophils, monocytes, and macrophages. Okay? Remember, neutrophils are the garbage trucks. Their job is just to gobble up anything they see and then quickly die. Monocytes and macrophages, however, are there to stick around for long periods of time, usually decades. And their job is to gobble up whatever they see, but also present or process and present those antigens to the immune system further. So as we said before, in the blood they are known as monocytes, and as soon as they leave the tissue, they then differentiate into macrophages. So how exactly does phagocytosis occur? Okay? And surprise, surprise, it's all based on chemotaxis, or following a chemical signal. And these chemical signals are often what we call PAMPs. These are pathogen-associated molecular patterns. And all that means is they are molecules that are found only on bad guys, okay? Bacteria, viruses, parasites. Now these molecules aren't very specific, they're very general, but they're enough to let the cell know, hey, this is bad, I'm going to eat you up and get rid of you. An example of a PAMP is LPS, lipopolysaccharide. Okay? This is a molecule, it's only found in gram-negative bacteria, so when a phagocyte sees this, it goes, you're bad, I'm going to kill you. Okay? These PAMPs bind to receptors on the surface of the cell known as PRRs, pattern recognition receptors, also known as toll-like receptors. And as soon as this PAMP binds to that receptor, the macrophage or phagocyte goes, oh, I'm beating you up. Okay? So the next step is engulfment. This forms a little compartment within the cell that has the organism in it, known as the phagosome. This then fuses with the lysosome, and this then destroys whatever is inside of the phagosome. Once it's destroyed, it's broken down into small little bits and pieces, which the cell then poops out. So here's an example. Up here we have a macrophage. You see all the bacteria. He's like grabbing onto each one of these guys and ingesting them. How does he know who to grab? Well, these bacteria have PAMPs on them. In this case, it's probably LPS. This binds to a receptor on the surface of the cell, and the cell goes, oh, I'm going to take you in, swallows you up. Okay? This then forms a phagosome inside of the cell. This fuses with another little lysosome, and this then releases a bunch of nasty things inside of the phagosome. By nasty things, I mean reactive oxygen species, or really catalytic enzymes, which literally chew up and break apart whatever is inside of that phagosome. This destroys the bacteria, and eventually turns it into small little bits and pieces, which can be eliminated as waste. Okay. So toll-like receptors, these are those receptors on the surface that recognize PAMPs, okay? Or these pathogen-associated molecular patterns. There are 12 toll-like receptors that we know of, and each one of them recognizes a different pattern associated with certain pathogens. So toll-like receptor 4, for example, binds to LPS, and this lets the macrophage know, hey, this is a bacteria, swallow it up. Toll-like receptor 6, I believe, binds to double-stranded RNA, which is typically only found in viruses. So if it sees double-stranded RNA, it gobbles whatever has that on it up, and then eliminates it as waste. So that is phagocytosis. Now a little segue, we'll talk about fever. Okay? I don't know why your book puts this here, it'd probably go better in the inflammation side of things. And that's because fever is where you elevate your body temperature, and this is usually brought on by systemic inflammation. Okay? What occurs here is your brain tells your primarily skeletal muscles to go into metabolic overdrive, and they start going crazy, releasing a lot of its energy as heat, and this elevates your body temperature. Okay? This is done by your body for a couple reasons. First and foremost, it's going to inhibit organisms that are very temperature sensitive. So a lot of true pathogens like to grow at exactly 37 degrees. So if your body pushes the temperature up to like 38 degrees, it puts them at a growth disadvantage. Okay? Also, because your body is going into metabolic overdrive, it preoccupies your red blood cells because they're going all over the place very, very rapidly. And what that means is that there's less iron available for bacteria. Bacteria need iron to survive, and the main way they get bacteria from your body is through blood cells. So if you use your blood cells up, keep them busy, it's less likely there'll be food for bacteria. And then last but not least, because the definition of fever is increased metabolic rate, that means your immune system will also have an increased metabolic rate, meaning it will work faster and better. Okay? And this is why when you have fever, you tend to get really tired, because your body is doing all this metabolic stuff, meaning you can't do other things like move around and kind of run around like a crazy person. All right, so that's enough about fever. Now we're going to spend the rest of this chapter talking about antimicrobial proteins associated with the innate immune response. So the first one is going to be interferons. And we've talked about interferon before briefly. And these are essentially proteins that are released by cells whenever they become infected by something. And they're designed to kind of warn neighboring cells that, hey, there's a problem, get ready, prepare yourself. There are three main classes of interferons: interferon alpha, beta, and gamma. Okay? And these produce responses to viruses, immune products, and various antigens that are inside of the cell. So these are important for intracellular infections, whether it's a bacteria that goes into the cell, a virus that goes into a cell, or a parasite that goes into the cell. So here we have an example. We have a cell. It's becoming infected by a virus. As soon as this virus gets into the cell, it turns on the interferon gene. This causes the cell to make interferons, which are released out into the environment. These then can bind to neighboring cells that aren't infected yet, and this prepares them for the infection that's coming, and they can develop antiviral factors that literally block the virus from causing an infection. The other set of innate antimicrobial proteins are complement proteins. Complement proteins are a series of 26 blood proteins, so they're circulating around in the bloodstream. They're produced by the liver, and they essentially work to poke holes in bad cells. Okay? And they can do this in one of two ways, using classical pathway or the alternative pathway. Now, the way the complement works is relatively complex. There's a lot of chemistry involved. I don't expect you to know any of that. But the two main pathways are going to be classical and alternative for us. And I do expect you to have kind of a rough idea of what's happening in each of those pathways. Okay? So we'll start off with the classical pathway. And the classical pathway requires an antibody. Okay? So we talked a little bit about what antibodies are in lab. Okay? We'll talk more about what antibodies do in chapter 15. But these are Y-shaped proteins that bind to antigen. Okay? So if you have an antibody in your body, It's in response to a pathogen that you once had. And so if you get infected with that same pathogen, the antibodies can then bind to it. Okay? Now once these antibodies bind to it, complement, which is found in the blood, can come and bind and form a complex around those antibodies. And eventually this complex forms something called a MAC, which is a membrane attack complex. And this literally creates holes in the cell, and then the cell dies. So classical complement requires an antibody, and once that antibody's there, the complement proteins form around and poke a hole in the cell. Alternative complement is a little bit different. So instead of requiring an antibody, it requires that the cell does not remove it. Okay? So alternative complement is in our blood, and all of our cells are exposed to it on a regular basis. But our cells know that it's there. So as soon as it lands on one of our cells quickly get rid of it. Okay? If you're a bacteria or a fungus or some sort of potential pathogen, you don't know this. So when alternative complement lands on your surface, you ignore it. And then all these guys, all these complement proteins can come, form a complex around it, poke holes in the cell, and then kill the cell. So classical requires an antibody, alternative requires you ignoring it, okay, and not removing it. And that is it for chapter 14. Any questions? All right, have a good weekend. Oh, really? You might be allergic to it.